Welcome back on my channel. We will continue packaging of the DNA helix, right? और ये रहा फिगर, okay. Nucleosome. Taking the distance between the two consecutive base pairs as 0.334 nanometer, okay. If the length of the DNA double helix in a typical mammalian cell is calculated simply by the multiplying the total number of BP with distance between two consecutive BP, that is 6.6 into 10 to the power 9 BP multiply 0.34 into 10 to the power minus 9 meter per BP, right? Sorry, it comes out to be approximately 2.2 meters, a length that is far greater than the dimensions of a typical nucleus. Okay, now we will see if the length of the E. coli DNA is 1.36 millimeter. Can you calculate the number of the base pair of E. coli? Okay, so now we will continue. In prokaryotes such as E. coli, though they do not have a defined nucleus, the DNA is not scattered throughout the cell, right? DNA, being negatively charged, is held with some proteins that have positive charges in a region termed as nucleoid. The DNA is nucleoid is organized a large loop held by proteins. समझ में आया कि नहीं बताएं? Right. So next we will see in eukaryotics. This organism is much more complex. There is a set of the positively charged basic proteins called histones. Okay. Core of the histone molecules, right? A protein acquires a charge depending upon the abundance of amino acids residue with the charged side chains, right? Histones are rich in the basic amino acid residue lysine and arginine. Okay. In their sides chains, histones are organized to form a unit of 8 molecules called histone octamer, right? The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome. Okay, nucleosome. A typical nucleosome contains 200 BP of a DNA helix, right? Okay. Nucleosome constitute the repeating unit of the structure in nucleus called chromatin. Thread like a stain, that means colored, right? Bodies seen in nucleus. The nucleosome in chromatin are seen as the beads on a string structure when viewed under electron microscope. Okay, EM picture. Okay, beads on a string, right? Next we will see theoretically how many such beads nucleosome do you imagine are present in the mammalian cell. The beads on a string structure in chromatin is packaged to form chromatin fibers that are further coiled and condensed at metaphase stage, right? Okay. Of a cell division to form chromosomes, the packaging of the chromatin at a higher level requires additional set of the proteins that collectively are referred to as non-histones commercial proteins. In a typical nucleus, some regions of the chromatin are loosely packed and are referred to as euco- euchromatin. Euchromatin. Euchromatin, sorry. The chromatin that is more densely packed and stains, more densely packed. Okay. Dark are called the heterochromatin. Euchromatin is said to be transcriptionally active chromatin, whereas heterochromatin is inactive. ठीक है, ध्यान दीजिएगा। एक्टिव में ट्रांस ये रहेगा, ये वाला, और हेट्रो वाला इनएक्टिव। सीधे पूछ देगा और पूछेगा क्या एक्टिव है कि इनएक्टिव, MCQ में। राइट, समझ में आया तो लाइक करें, सब्सक्राइब करें। थैंक यू फॉर वॉचिंग।